I wasn't going to make a video on meiosis because I found some pretty good animations online, and I've posted those links on Blackboard. But some students have requested a recording on meiosis because they say that the recordings sometimes can be very helpful. So I'm going to focus on sexual reproduction and meiosis in this lecture recording. So sexual reproduction, specifically meiosis and fertilization. You already know what fertilization is. And in here, meiosis produces gametes. If you remember, that's sperm and egg, right? So the gamete will go through fertilization, and that will produce a new cell called zygote, and zygote develops into embryo and eventually a baby. So sexual reproduction introduces variation into the offspring. And why is it important? Because this accounts for the evolutionary success of those organisms. Why is that? Now, we know that mitosis is pretty much just cloning the parent cell, right? So the daughter cells, the new cells, at the end of mitosis are exactly the same as the parent cell. So, sorry, I don't have too much space here. So the daughter cells are identical to the parent cell. So we can call them, clones of the parent cell. Now, this is okay if there's nothing changing in the environment, right? So if the parent cells or the parent organisms can survive and reproduce and make offspring, that means they're pretty successful. But there is a challenge. If you think about it, everything in the environment constantly changes, right? So whether it's abiotic factors, abiotic components like a temperature, humidity, the amount of water in the environment, those things change all the time. And also the biotic components, like the organisms that you interact with, like your predators or your prey, they change all the time too. So if the environment is changing, then in order to adapt to it, that means you have to have some variations in the offspring, right? Because you don't know that maybe by chance one of these variations will produce some favorable traits that make you more successful surviving and reproducing in the environment. All right. For some reason, my pen is not working. Okay, got it to work. So we talk about the significance of meiosis and having variations in the offspring. For meiosis, remember this, that the starting nucleus is always diploid. And then at the end of the meiosis process, the new cells are going to be haploid. Okay? So for humans, we have different cells that will make either sperm cells or eggs. So the one that makes sperm cells is called spermatogonia. So spermatogonia is diploid. So that means the cell has 2n, in terms of chromosome numbers, right? So for humans, n means 23. So that means you have 2 copies for each chromosome, one from the mother, one from the father. Now, the spermatogonia is going to undergo meiosis, and it's going to make sperm cells. Okay, so sperm is, it's haploid. Sorry, I don't have any space here. Haploid, so that means sperm cells only have n for chromosome numbers. For females, we have oogonia, which is also diploid. And the oogonia will go through meiosis, and it's going to produce oocyte, or we can just call it egg. And the egg is haploid and only has n in terms of number of chromosomes. All right, so remember that. To achieve the reduction by half in chromosome number, the meiosis consists of 1 round of chromosome duplication. This is exactly the same as mitosis, right? In mitosis, you also have a 1 round of chromosome duplication. But the difference is how many times the cell divides. So for meiosis, the cell is going to divide twice. Okay? And in mitosis, only once, right? So the number is the same. The number of chromosomes remains the same. But for meiosis, the cell is going to divide twice, so you want to have half the number of chromosomes as the parent cell. So this is a quick overview of meiosis. Now, because there are two divisions, so we are going to call the first division process meiosis 1, and sometimes you see this number, the Roman numerical, and then meiosis 2, so that's the second division. All right, now if you look at this picture, you start with a diploid cell, right? So 1, 2, 3, 4. So you have 4 chromosomes in total. So you have 2 different types of chromosomes. And then for each type, you have 2 copies, one from the father, one from the mother. So 2n is 4. And then at the end, you can see that you make 4 daughter cells, and each daughter cell only has 2 chromosomes. So n is 2. So this is half the number of the chromosomes in parent cell. Okay. Now I just want to mention this real quick because this picture is pretty good. So in meiosis 1, what you are separating is homologous chromosomes. Homologous chromosomes. This is where you are going to split in meiosis 1. So if you don't remember what homologous chromosomes are, please do a search and figure out, because we did talk about this in the class. Remember, I asked you to count your fingers and then to look at your hands. So if you look at both hands together, that represents diploid, and then the 2 thumbs together, they are homologous chromosomes, and 2 index fingers, they're homologous chromosomes. Okay. So again, homologous chromosomes, same number of chromosomes, but different versions. One from the mother, one from the father. Okay, so let's say blue is from father, red is from mother. Now, in meiosis 1, we'll talk about details in the next few slides. Eventually, you are going to have these homologous chromosomes pair up. See, they pair up. All right. And then they are going to line up near around the metaphase plate. Okay, so this is the metaphase plate, and you can see the homologous chromosomes line up on both sides of the metaphase plate. And then at the end of meiosis 1, you are going to separate the pairs of homologous chromosomes. So that means that these 2 will be separated, and these 2 will be also split. Okay, and then at the end of meiosis 1, you end up with, right, if you separate it this way, a big red and a small blue, and then when you look at down here, you have a big blue and a small red. So remember this is very important. In meiosis 1, you are splitting the homologous chromosomes. Okay? All right, now this is what you have in meiosis 2, and from here to the end, this is pretty much the same as mitosis. So this is what you are more familiar with. Okay? So remember what happens in mitosis? You have these sister chromatids, right? So these are called sister chromatids. And in metaphase, they line up in the middle, right? And then in the next stage, anaphase, these 2 sister chromatids are split, and then they end up in 2 new cells. So these used to be 2 sister chromatids, and these used to be 2 sister chromatids. All right. So this is the kind of general quick overview of meiosis. All right. I have a few pictures here, so you can find whichever one that works for you. So this picture is very similar. I like this picture because it has a different number of chromosomes. So I'm going to ask you to tell me how many chromosomes do you see in the parent cell. So let's count. 1, 2, 3, 4, 5, 6, right? So that's a 6 total. And that's a diploid cell. So 2n is 6. So that means n is 3. So you have 3 different types of chromosomes, and each one has 2 copies from the 2 parents. All right. Now in metaphase 1, the 3 pairs of homologous chromosomes are going to line up, right? So you can see this is one pair, this is one pair, this is another pair. How do you know? Because they have similar shapes, they have similar sizes, right? And then in anaphase, these two homologous chromosomes will be separated. There you go. And then you can see the two daughter cells at the first round of a division. Each one has how many chromosomes? 1, 2, 3, right? So that's 3 chromosomes. But how many sister chromatids do you see in each daughter cell? So 1, 2, 3, 4, 5, 6, right? So total is 3 chromosomes, but it's actually 6 sister chromatids. Now, I remember I told everybody that if the 2 sister chromatids are still attached, like they're still one piece, we count that as a 1 chromosome, right? So since they're still attached, we count that as a 1 chromosome. So 1, 2, 3. So that's 3 chromosomes. But you need to know that it's actually 6 sister chromatids. Okay. All right. Now, in meiosis II, like I said, that's pretty much just mitosis, right? So for each chromosome, you already have 2 copies, the duplications, which are 2 sister chromatids right here. And then you are going to have everybody line up in the middle, right? And you are going to split the 2 sister chromatids. Okay. So now at the end of the meiosis II, each cell has, this is a little bit hard to see because it indicates that this is chromatins, because the chromosomes start to decondense and they go back to chromatins. So for each cell, you can see 1, 2, 3, right? So each daughter cell has 3 chromosomes. And how many do you start with? 6, right? So it's only half the number of the chromosomes in the parent cell. Okay, now the next few slides have some details about each stage. Okay, so I'm just going to point out the important things. So prophase I, make sure you know that this is prophase I, because in each cell division we have a prophase, metaphase, anaphase, telophase. So we are going to assign everybody a number. If this happens in meiosis I, then we're going to call it prophase I, metaphase I, anaphase I. If it's meiosis II, the second division, then everybody will have number 2. Okay, so in prophase I, homologous chromosomes come together to form a synapse. So the synapse really just means the homologous chromosomes pair up. Okay, now if you think about this, let's say this is the nucleus, right? So you have the cell, and then this is the nucleus. Usually in the nucleus, you have chromosomes. Now they have already replicated at the end of interphase, right? So I'm going to draw the X. Usually they just kind of, randomly just kind of floating in the nucleus, right? But in prophase I, the homologous chromosomes will come together, okay? So there we go. These 2 are homologous chromosomes, so they will come to each other. This is another pair, and this is another pair, okay? So this is what happens in prophase I. And these chromosomes are bound tightly together and in perfect alignment. So this is what you see, right? So they're aligned almost perfectly. Okay. A couple of terms. Don't forget, this is centromere. It's almost like you try to kind of pinch the chromosomes, the 2 sister chromatids. They have like a very narrowed part. So this is centromere. And kinetochore. So that's the proteins that can kind of bind the centromere to the mitotic spindles, right? So this is how you can separate things. All right, and then we already talked about that these are 2 sister chromatids, the 2 red ones, and then the 2 blue ones, those are another pair of sister chromatids. Okay. During prophase I, again, this is a very important concept. Crossing over, or a lot of times we'll just say crossover, occurs during prophase I. What is crossover? Now remember in the previous picture that the homologous chromosomes, they're almost in perfect alignment, right? Let's say we have these 2 homologous chromosomes, okay? And we have different genes, let's say ABC, and because they come from different parents, so we're going to say for the father, it's one version of those genes, which is going to be indicated by the capital letters, okay? And then for the one from the mother, we're going to use the lowercase letters, okay? So there we go. And when they line up, they may exchange part of the chromosomes. So you can see here, they almost kind of link their arms, right? So they can exchange the genetic material. So what happens is the blue one is going to get, the segment from the red chromosome. So instead of a capital C, now you have a lowercase c. And then it's the same for the chromosome from the mother. Like this arm of the sister chromatate has the blue segment. So instead of a lower letter C, you have a capital letter C. Okay, so this is called crossing over. Now, you can probably guess that crossing over can contribute to genetic diversity, right? So instead of blue, you have a segment of a red, right? So you have something different introduced. So that will increase the genetic diversity. So there